do infection imaging and some tumor imaging with it. And if you've noticed in the chapter that we have for the requisites, I think they call it inflammation and tumor imaging. Okay. So basically you'll see that gallium is used for both infection imaging, mainly chronic type infections. Indiamoxine is what we use to label white blood cells. By the way, you guys have all done white blood cell imaging, correct? Okay. Normally all you do is you collect about 60 cc's of blood, or sometimes less. Yeah. How much do you collect?
60.
Yeah. Then, normally what they do is they spin it down, they wash it. They do it a couple of times, mainly because indium not only tags the white blood cells, but it also tags platelets. So you don't want to get confused, and if you see an accumulation of indium, you don't want to think that it's an infection when it's just actually a blood clot that's forming. So normally they'll wash it, they centrifuge it, they remove the white or the red blood cells, they wash them down again, and then finally when you get through spinning it down, you're left with what they call a button. It's just like a little white blob down at the bottom of the test tube, and that's your white blood cells. It's what they call the Buffy coat. So we just put drops of indiamoxine on there, and it gets taken up by the white blood cells. A few years ago, they started using technetium HMPAO. Remember that we normally use that for brain imaging. But if you don't put the methylene blue preservative, you can label white blood cells with it. So the nice thing about this is that you can usually prepare it pretty quickly. Typically they use it for imaging things like appendicitis. Because when we start talking about white blood cell imaging, anytime you inject this stuff initially into the patient, there tends to be an accumulation in the lungs. For the first few hours, the stuff accumulates in the lungs, and then it starts, diffusing into the rest of the body. So basically, if you were looking for anything like a pneumonia, you wouldn't be able to see it in the first 2 hours. And then what's the problem with HMPAO if you wait longer than a few hours?
It's not right, 30-minute half-life?
No.
Isn't it done in 30 minutes?
How does HMPAO get cleared?
Kidneys?
No, not the kidney. Liver, yeah. So what happens is, with HMPAO, the reason why, it's not good for delayed images, especially the abdomen, is because it's cleared out just like a HIDA scan. So the thing is, if you're looking for an infection in the abdomen, you can use HMPAO, you can inject it, you can image pretty quickly, but then after 2 hours, pretty much you can't use it. That's why indiamoxine is used. You can inject it, but typically, when do you image indium? Yeah, usually it's 24 hours. You do a 24-hour scan, maybe a 48-hour scan. And the thing is, by that time, the stuff has left the lungs, and because indiamoxine does not leave through the liver, you're able to get pictures of the abdomen. So
indiamoxine collects in the lungs as well?
Yeah, initially it does. So normally for the first couple of hours, taking an image of the lungs is kind of a useless activity, because both of these will have high concentrations in the lungs. But if you needed to take a picture of something going on in the abdomen, especially very quickly, then HMPAO would be your choice. So if you're looking for like appendicitis or diverticulitis or something, if you needed to image it quickly, then HMPAO would be the way to go. But after 2 hours, then you start having a problem with stuff that's been excreted out through the biliary system. So you can't use it. The other thing here, albumin colloid and sulfur colloid are also used for inflammatory imaging. I think Greg asked me a question last semester about doing a sulfur colloid and an indium scan. Okay? Because sometimes, do you remember what the rationale was? Greg, since I put you on the spot.
I think, wasn't it like a tumor in the liver, and it would be not sure what you're asking.
Okay. Sometimes you may do a technetium sulfur colloid scan as well as an Indian white blood cell scan because sometimes you have bone marrow, increased bone marrow activity. So the sulfur colloid will be active in that area. And so if you have white blood cells and sulfur colloid both active at the same time, that means that you probably don't have an infection. Okay? What they call concordant scans. But if the Indian white blood cell scan is hot, the sulfur colloid scan is normal, then you probably have an infection. So we're going to talk about that a little bit later. So any of the albumin colloid, if you remember that sulfur colloid and albumin colloid all go to the liver, spleen, and bone marrow. Okay? So sometimes you can have that. One of the things we're going to be talking about here, okay, there's certain responses that allow white blood cells to respond to an infection. And so what I listed here first are what we call the vasoactive chemicals. Okay. One of the big ones is histamine. We also know that histamine is a powerful vasodilator released by mast cells. Does everyone know what a mast cell is? These are like precursors to your, they're like the main cell for, for your immune system. But the ones that we're looking at here, the mast cells that usually create problems for us are the ones that release histamine and cause allergic reactions. Okay? If you have a sensitized immune system and you release histamine, then you can have some problems and go into anaphylactic shock. So notice that so they produce histamine, which causes a sensitization of your immune system. Usually when you have an oversensitization is when you have the allergic reaction. But this is what gives you the hives, the swelling shut of your respiratory system so you can't breathe, and things like that. Then I have bradykinin here. Notice it says stimulates dilation of the surface blood vessels near the sweat glands. So this is why when a person has a fever, and when this kicks in, it allows the body to sweat and cool down. Because usually when people have a fever, initially they have a high temperature, but they're not able to cool themselves immediately. So bradykinin allows the body to release heat to keep the body temperature down. Okay. Because you guys have all remember, did any of you guys when you were kids have your mom call 911 because you weren't breathing? Because you had a high fever? Yeah, in the sheriff department, in November, December, when the flu and cold season comes in, we get all sorts of calls of babies having seizures and stopping breathing. So moms and dads don't know, but basically what happens is their body temperature gets so high and the hypothalamus kicks in and resets the thermostat, so cools them down. And remember, the hypothalamus not only keeps track of your hormones, but it also helps regulate your body temperature. And so when a child has a seizure, typically that's what's happening, is that body temperature is so high that it's affecting the hypothalamus, so they start having seizures. But anyway, in this case here, this is what helps cool you down when you have a fever. And then we have serotonin, which allows white blood cells to come in. This is a vasoconstrictor, inhibits blood flow to the injured area. It's released by platelets to stop bleeding. So you have a combination of things going on at the same time here. You have the histamine, which down here it's increasing the permeability, so white blood cells can go in and attack the infection. You also have fluids that are going in and out that are causing swelling and putting pressure on blood vessels. We also have what they call a release of chemotaxic agents. You ever heard of that one before? Chemotaxic drugs or agents are things that call white blood cells to a certain area. It's kind of like when a shark smells blood. If there's blood in the water, it kind of draws all the sharks to that area. Well, chemotaxic agents do the same thing, except they cause white blood cells to be drawn to the area where the infection is. And then over here you have redness, warmth, and swelling. Okay, these are, if you remember the, in anatomy and physiology, they had a thing for that. It was like erythema, edema, and algia. Algia for pig. Okay, but these are the things that let you know that you have an infection going on usually. Going back to your good old days of anatomy and physiology, if you remember, there are basically two types of cells that are produced by the bone marrow that are responsible for fighting infection. You have the granulocytes and the agranulocytes. Okay? This is important because some of the questions that you're going to have are, which types of blood cells do you actually label? Okay? What white blood cells are we actually labeling? Granulocytes are basically going to be white blood cells that have what look like little particles in them when you stain the cytoplasm. Okay, the agranulocytes, when you stain the white blood cells, there's nothing there. They don't pick up anything. And then these other things here, the neutrophils, eosinophils, and basophils, these are dyes that are picked up by certain white blood cells. So, for example, a neutrophil is a white blood cell that doesn't pick up any of the dyes that we normally use when we stain white blood cells. Did you guys ever do that in your anatomy and physiology, stain white blood cells? Okay. They put this like blue dye in there, and then you can see all the cool stuff. And so neutrophils don't pick up anything. That's why they're called neutrophils. We have a dye called eosin, and if the white blood cell picks up that, then it's an eosinophil. Normally we see these in parasitic infections and allergies. Okay, so that's one of the reasons why when you do a differential CBC, they'll actually identify, like, you know, how many red blood cells you have, how many immature red blood cells, how many neutrophils, eosinophils, and basophils, things like that. Okay, then the basophil. Again, there's some of these blood cells we don't really know what they do. And I put down here other things like the lifespan. Okay, these are important for a couple of things. Again, when you label these, you have to remember that they're very delicate. So one of the things that they do before they send your white blood cells back to inject in the patient, they have to make sure they didn't kill too many of them. I don't know if you're aware of that, but labeling white blood cells, you actually have to do like a survival test on them to make sure that there's enough of them left to actually do some good for you. Okay? But notice that they usually have like a very short lifespan, 7 hours in the blood and 2 to 3 days in tissue. It's not very long, is it? Eosinophils, a little bit longer. And then the basophils, again, they don't know, how long they are, but again, they do release certain substances. The heparin and the other types of things are designed, again, to allow white blood cells to more easily penetrate through the tissue to attack the bacteria. Okay. Now on the next page, these are the ones that you're probably more familiar with. The agranulocytes. Notice much longer lives. Again, some of these things are the ones. These are the ones that are normally responsible for your long type of immunity.
Can you darken it up a little bit?
Is it doing anything?
Mm-mm. There.
Is that a little better?
Yes. Thank you.
Okay. But again, the granulocytes are the ones that we're also concerned with for a different reason. They're not really involved that much in infection type processes, but the lymphocytes especially, these are where we get our antibodies from, and so when we start talking about monoclonal or polyclonal antibodies, this is where we get them from. Okay. So when we start talking about that. But what I started out with here, the B lymphocytes, these are the ones that most people are familiar with. Notice it says that they were called B lymphocytes because they're typically found in areas of the body that in constant circulation. The other ones, the T lymphocytes, are the ones that are found in the thymus gland. And if you remember, the thymus gland is one of our endocrine glands that secretes what hormone? Okay, thymosin. And thymosin is responsible for maturing your immune system. Okay, so anyway, so we've got the plasma cells, and they usually go to other areas of the body where they hang out and mature. And I said, when you're looking at the percentages, when they're doing a breakdown of the differentiation or differential, this is important because this is how we diagnose things like HIV. Okay, but these ones here, the B lymphocytes are the ones that are normally responsible for producing antibodies. So basically, when they detect a poison or an infection or something, they'll start producing antibodies or immunoglobulins that will attack. Normally they don't do things other than cause these foreign proteins to clump up so that the phagocytic bacteria can attack them and more easily get rid of them. Okay. We also have lymphocytes that will grab a foreign protein, take it to a lymph gland. The lymph gland identifies what type of a protein it is, starts making antibodies against that protein, and then manufacturing them. Okay. So that's what I put down here. So the B lymphocytes, they'll find a receptor on the antibody. Now, does everyone, if you remember, we have what they call an antigen, which is the foreign protein. We have the antibody, which is the cell's response to that protein. In other words, it identifies that protein and then manufactures something to counteract the effect of it. And so what I listed down here, okay, so we have, this is the T-dependent. This is where one of the thymus gland cells, the T helper cell, will bring that foreign protein to the lymph gland and have it identified. I put down here that there's another thing called a cytokine. I don't know if you've heard those, which sort of turns on these things. When we start talking about cancer, you're going to hear this again, because one of the things that they think causes cancer are some of these enzymes or proteins that turn on and turn off cells. In other words, cells are constantly emitting these different substances, and in this case here, this one turns on the lymphocyte, but then we also have to be able to turn it off, because otherwise they get too aggressive and they start attacking everything. So on this thing here, cytokine is one of the things which turns on the B lymphocytes, and that will start the manufacturing process of antibodies. Then I put down here the definition of an antigen. And so this is what triggers your immune response. I put down antigens elicit a response due to the epitope. And this is what we're talking about here. If you have an antibody, this is the IgG. At the end of the antibody is the region that is stimulated by the epitope. And so this is what starts that chain reaction. Okay. You will find out that immunoglobulins, we have a whole bunch of them. And this is the one that we use for monoclonal antibodies, the IgG. But there's also IgA, IgM, if we were looking at your body to find out if you had an allergic reaction, we would be looking for IgE. But each one of these different immunoglobulins is produced in response to a different stimulus. So sometimes they do electrophoresis. In other words, they'll take your plasma, they'll put an electric charge on it, and then these separate out according to their weight. So the IgG is the smallest one, and again, so this is the one that we use for most of our monoclonal antibodies. Okay. The bad news is that we don't really, well, actually I shouldn't say the bad news. They are starting to use antibodies for imaging infection. Most of the stuff that we do antibodies with right now is for tumor imaging. So, but anyway, so just keep in mind that IgG is the most common one. This other stuff here, I don't really care that you know this, but just there are some other different molecular weights of it. So there is IG1, IG2, IG3, IG4, and when they're doing a complete analysis of your immunoglobulins, then they'll be looking at that. Sometimes if you get exposed to hepatitis, they'll probably give you an injection of IgG. That's what they used to do for any type of needle stick that you got. Okay. The other thing here, this down here, the reaction called complement fixation. This is what occurs with the immunoglobulin reacting with the foreign protein and then allowing it to be destroyed by your neutrophils, for example. So this complement fixation occurs. You get the proteins, the antigen and antibody, they lock together. That neutralizes the ability of that foreign protein to do any damage. And then what happens is the proteins will then become lysed. So in other words, the antibody will start breaking down the cell membrane, and then it causes the release of histamine. Okay, the histamine is going to stimulate the other cells in your immune system, the B lymphocytes, also the neutrophils. And then it says the chemotaxic agents are then released to attract leukocytes. And then the combination of the antibodies sort of clumping all these foreign proteins together and then allowing your neutrophils to attack them and digest them, that's the overall effect of the IgG on that. Kind of cool, huh? The other thing that I have down here, okay, now you will see this IgG again and again because almost every single any monoclonal antibodies that we use are formed using IgG. Does everyone remember which are antibodies that we use? Yeah, anything that has MAB at the end of it, Capromab or anything like that. Does everyone know what that stands for? Mouse antibody. Yeah, so Capromab or anything like that. If it has MAB at the end of it, then basically it's an antibody that's used for imaging. The interesting thing about this is, IgG is one of the smaller immunoglobulins that we use. That I put down here, it has a weight of 150 Daltons. I have no idea what a Dalton is. Okay, but that's how much it is. I can tell you that the other ones are much bigger than that. So this kind of controls how well the immunoglobulin is able to go in between the cells, like intracellular or intercellular. And in fact, what we do when we make antibodies, we actually break these things up and make them smaller. Sometimes we cut off the bottom, sometimes we cut them in half. We create what we call fractional antibodies. So you'll see FAB or something like that. You should keep in mind that, okay, it looks like the letter Y. The ends of this thing are called the variable region, and this is where the recognition and antibody-antigen reaction occur. Okay, And there's two sites for that. We have a thing called a short chain, which is a protein band along the side of it. Then we have the long chain. The long chain is this big, long one like that. And then down at the bottom there's a thing called a constant fragment. It doesn't, it's not something that we use. In fact, sometimes we cut this off. Okay, we have different enzymes that we can use to either cut this off or to divide the antibody in half, so then we get twice as much for it. You should know how antibodies are made. Okay, basically, now I know that doesn't exactly look like a mouse, but maybe someday Mr. Radke will get art lessons to...
That's a lot of rabbits.
Well, yeah, it's supposed to be a mouse. Actually, it's called they actually, from my understanding, it's a Chinese hamster, but they call it a mouse. So I don't know. But basically, if you take a tumor, let's say you have a cancer like a prostate cancer, and you inject it into a mouse, because it's a foreign protein, the mouse is going to form antibodies against it. When they're trying to create antibodies, what they typically look for are animal species that are very different from each other. So, for example, they're not going to use like a rhesus monkey or a chimpanzee to make any human antigens because they're too close to ours. So the more foreign the protein is, the better the antibody response. Okay, that's the idea behind this. Okay. So you inject the tumor into it. The mouse forms antibody against it. Unfortunately for the mouse, we sacrifice it and we take out its spleen. Okay. So the spleen is where we isolate the cells producing antibodies against the protein that we injected. Okay. So that's why I said we take splenocytes here. If we were to look at the serum in the mouse, we can't use that because any other contaminants that are in that when we injected it into the patient are also going to form antibodies. And that's why they call polyclonal antibodies. So those are things that you cannot use in the development of an imaging radiopharmaceutical. Does that make sense? So they're not going to take the animal's blood and spin it down and look for things like specific antibodies against it because there's too many contaminants. If you've ever gotten a tetanus shot, back in the old days of tetanus shots, they used to use horse serum. It hurts, doesn't it? But the problem with tetanus shots in the past with horse serum was they would take the tetanus bacteria, they would inject it into the horse. Actually, they would take the tetanus toxin, inject it into the horse. The horse would form antibodies against the toxin, and then they would isolate that and inject it into people. Okay, the bad news was that because of the horse antibodies are a foreign protein, the second time you got a shot for tetanus, it would either kill you or your own immune system would destroy those proteins. And then this is one of the problems that we have with monoclonal antibodies now, is that when you inject them into a patient, your body forms antibodies against that, and so sometimes you may have allergic reactions or you may have no reaction at all because your body has formed antibodies against these monoclonal antibodies, and as soon as they're injected into your body for a second time, they're destroyed immediately. So, anyway, so going back over here then, what we're looking for are the splenocytes, and we fuse them with multiple myeloma cells. Now, multiple myeloma cells are cancer cells of the bone marrow. Okay? In fact, multiple myeloma is a very horrible disease, but if you can take those and mix a spleen cell with this, you create a factory that's almost indestructible and will be producing monoclonal antibodies forever. Does that make sense? So in other words, multiple myeloma cells, very difficult to kill. You combine them with a spleen cell and you create this thing called a hybridoma, and then you got a factory. You've got a fortune, because if you can produce antibodies against certain cancers, then you're going to be making a lot of money. And this may be one of the ways that they treat cancer in the future. I mean, they're looking at it very carefully, and it seems to be very effective. Okay. So this is what I have here, and this is what I was talking to you about. The whole antibody looks like the letter Y, and whenever you see it, it's called an MAB, and that stands for mouse antibody. Okay. They typically don't use the whole antibody by itself, but what they do is they fractionalize it. And this is what this FAB means. Okay. To make it smaller, remember we told you that it was 150 Daltons? Okay. By using pepsin, and remember, where do you find pepsin at? Yeah, parentheses. It's a digestive enzyme. Breaks down proteins. So you take the antibody, you mix it with a little bit of pepsin, you remove the constant fragment, it makes it much smaller, so now it's able to penetrate into more difficult areas. Chief cells? Okay. If you ever see this, and sometimes you probably will, does this look like it's getting brighter again to you?
Mm-hmm. Yeah. Yeah.
Okay, so an FAB2 means that they just cut off the constant fragment. There's another enzyme called papin, and basically they split it down the middle. So this makes it about 75,000 Daltons, or 75,000 Daltons. And so that's a FAB prime. Okay. Like I said, when you guys are reading through and you're looking at all the different monoclonal antibodies that we use for, colorectal imaging, prostascent, all of those things, these will come back again to haunt you. Okay? So, these are some of the other monoclonal these are some of the other antibodies that are there. This first one that I have up here, the IgM, anytime you are exposed to a new foreign protein, this is kind of like a general response, immunoglobulin, and basically all it does is cause things to clump up. Okay. The IgG is the one that that's the one that's more specific for fighting things. Then I've got the IgA, and notice that those ones you're going to see in areas where you have body surfaces that are exposed to the environment. Because notice it's in the tears, saliva, bronchi, and intestinal tract. So any pathways that bacteria can use to enter your body from the outside, IgA is one of the immunoglobulins that are there. These are the ones that also, help dissolve bacteria and stuff from your tears. It's actually pretty good stuff. The IgE we already talked about. When people have allergic reactions to things, like if you've been stung by a bee, or you've taken penicillin and then you take it again, or you get stung a second time, if we were to look at your immunoglobulins, we'd see a high percentage of IgE in there. So going against other foreign proteins that you've already been exposed to. And also parasitic worms and things like that. It's one of the, like I said, if you could spend almost a whole course doing, immunoresponsive things. The other thing that we're doing with this too is, you've heard of immunotherapy for treating cancer? You haven't heard of that? What we do is we sensitize the immune system, and there are certain drugs that we give. Because if you remember, we had the cells called the T killer cells. Your body sort of has an immunosurveillance program where the T cells in your body, you have the T helper cells, you have the T killer cells. And what basically happens is that these cells go around and they identify your normal body cells that are changing. and when they recognize that it's changing, they attack them and kill them. The T killer cells especially, if they see that a cell is turning cancerous, they go over there and they inject it with hydrogen peroxide. Hydrogen peroxide is a great poison that we use to kill things. But anyway, so I just wanted to let you know that there are these other immunoglobulins, and I put down here that you know, in a lot of cases they don't know what these things really do, what their purpose is. I mean, so when I was trying to put this thing together,
Yet to be figured out.
yeah, you know, I mean, there's spending a lot of money doing research on it, but the thing is, for right now, we can't really tell you that.
Do certain ones like last a lifetime,
Mm-hmm.
and others not so long?
Well, yeah, there's some that the body keeps manufacturing to replace, and that's what we're talking about here with the memory cells. It says 10 days after the first exposure, antibodies can be detected. And this is kind of the bad thing because, you know, especially like if you've been exposed to AIDS or anything like that, it doesn't show up for a long time. And what we typically do with AIDS is, you know, remember, what are the 3 cells that we have that are considered T cells? We have the helper, we have the killer. What normally happens is that with AIDS, you wipe out the helper cells. And so that's why the body isn't able to fight infections anymore. The virus attacks those specifically, and so, but in this case here, so about 10 days after you've been exposed to something, you'll start seeing antibodies in there. And then it says, certain lymphocytes serve as memory cells, which means they remember a foreign antibody for a long time. This is what vaccines do. Okay, so if that particular bacteria or virus attacks you again, the body is able to manufacture antibodies against that specific protein more quickly. Okay, so that's kind of what that's for there. Let's see what else we have here that's interesting. Okay, this is our one little section about the T lymphocytes here. Okay. So again, keep in mind that the T lymphocyte helper cells are the ones that deliver foreign proteins to the B lymphocyte so they can start manufacturing antibodies. The suppressor cells, that's the one I was thinking of. The suppressor cells are the ones that turn off your immune system. Otherwise, your body would be making, you know, spending too much energy defending itself even after the threat of the bacteria is gone. I don't know if you're familiar with this, but you know, people who get cancer and other types of diseases, they become very, what they call cahexic, they don't have any, they're starving to death, literally. You know, a person who has cancer, one of the reasons why they lose weight is because the cancer cells are metabolizing things more quickly than normal cells. And basically, a person can't support the calories to keep their body going. You know, it's like when a person has a fire, is burned in a fire. The amount of repair that needs to be done and the amount of energy for the body to apply to that repair process isn't available because you can't eat that many calories. You know, a burn victim typically requires about 8,000 calories a day. And most of us only take in about 2,000. So a lot of burn victims, they not only die from infection and loss of fluid, but literally they starve to death because their bodies just don't have the ability to metabolize that, the amount of food that they need to eat. So the suppressor cells here turn off your immune system because otherwise if you don't, then you start attacking yourself. Remember that there are some immune diseases, you know, Hashimoto's thyroiditis, rheumatoid arthritis, rheumatic heart disease. These are situations where the body took care of the toxin that it was fighting, but rather than turning off, it continues to attack and starts going after your cartilage. Remember, there's streptolysin, that's produced by the strep bacteria. And every time your body gets an infection to that, it sometimes tends to overreact, and that's why it starts attacking itself. These are the nasty ones here, boy. If you want a real hitman, the cytotoxic cells, when they detect a cancer cell, well, I mean, these guys go after mostly the bacteria and the viruses. These ones down here, the natural killer cells, these are the ones that go around, and when they identify that one of your own body cells is becoming cancerous, they kill it. Okay? So we've got the cytotoxic ones, and we've got the natural killer cells. That's what I want to be when I grow up. Go around there and inject those. Okay. Then down over here, let's see what else. The lymphokinins. These are the types of materials that dissolve the protein. They also act as a chemotoxic agent, where they'll, or chemotaxic agent, where they call other cells over to help fight the bacteria. And I've listed them here. These are also things that we suspect cause cancer. Okay, they stimulate cells to grow, and that's why we have the monocyte macrophage colony stimulating factors. So these are things that tell the cell to turn on. We also have, I don't call them proteins that tell the cell to turn off. So all of these things here are enzymes and proteins that cause the cells in that area to start reproducing themselves and being attracted to an area where there's an infection going on. Sound good so far? Okay. Now, the other thing down here, I wanted to sort of hold off before we start talking about these monoclonal antibodies, because most of these things here are not used for infection imaging. Okay. The OncoSynth. Okay. Has anyone used that before? Ashley saying no? Okay. Basically on this one here, we're looking for colorectal cancer and ovarian cancer. But we'll talk a little bit about that in just a second. What I am going to do, though, is we're going to switch gears here. And I will send you a copy of this. Okay. So these are the types of things that we're going to be discussing. The white blood cells labeled with either technetium or indiumoxine, gallium, and then, like I said, they've come up with antibodies now that they're using specifically for an infection, even though right now most of the antibodies that we've discussed this morning are the ones that we use typically for tumor imaging. And then, of course, we've got good old FDG. Okay. By the way, you guys want to take a short stretch? Okay. I'm going to take about a 10-minute break.